Hi, my name is Andrea Navarrete Vargas. I'm a third-year student majoring in cell biology. Over the past year, I've had the amazing opportunity of working in the Dr. Borodinsky lab located in the Institute of Pediatric Regenerative Medicine at Shriners Children's Hospital. The project I will be presenting today focuses on how cold temperature influences motor neuron survival. Specifically, how trypamate activity is necessary for motor neuron survival in Xenopus laevis embryos raised at cold temperature. Uncovering the mechanisms for motor neuron survival is important because motor neuron diseases are a group of neurodegenerative disorders that are directly related to motor neuron degeneration. In the U.S., amyotrophic lateral sclerosis, or ALS, the most common of these diseases in adults, affects approximately 30,000 individuals, with 5,000 new cases each year. Motor neurons originate from the spinal cord to connect with the skeletal muscle, thus enabling motor function. Our lab uses the model system of Xenopus laevis, the African clawed frog. These embryos develop externally and can tolerate extensive manipulation, such as injection and dissection. Their external development also allows us to track their development of the spinal cord, as seen here. Previous studies in the lab demonstrated that animals grown at cold temperature exhibit higher number of motor neurons than siblings grown at warm temperatures. This cold temperature dependent increase in motor neuron number correlates with a decrease in motor neuron programmed cell death, also known as apoptosis. The mechanisms which contribute to the survival of motor neurons at cold temperatures remain unknown. The schematic on the right summarizes these findings. Here we are seeing a transverse section of the spinal cord, shown in green. We can see that there are more motor neurons in the spinal cord of those grown in cold temperature on the left compared to those grown in warm temperature on the right. We see that this is due to a decrease of apoptosis in embryos grown at cold temperature versus those grown at warm temperature, represented by the gray apoptotic cells on the right. Trypamate is a cold-sensitive ion channel that is activated by cold temperature, allowing calcium to flow into the cell, represented by the purple protein channel. I believe this channel is playing a major role in decreasing apoptosis in embryos raised at cold temperature. Therefore, my hypothesis is that trypamate is necessary for motor neuron survival in animals raised at cold temperatures. To test this hypothesis, Xenopus laevis laid eggs were fertilized in vitro with dissected testes. At the 2-cell stage, embryos were injected bilaterally with 5 picomoles of either trypamate morpholino or control morpholino. A morpholino is an oligonucleotide construct that blocks translation. Therefore, by injecting trypamate morpholino, we are blocking the translation of this protein. However, injecting control morpholino will just mimic the stress of injection. Using fluorescent microscopy, the following results were obtained. In these images, we are again seeing transverse sections of the spinal cord for embryos grown at cold temperature, being 16 degrees Celsius. We are looking at the anterior region of the spinal cord, closer to the brain. On the left, we have our control morpholino group, and on the right, we have our trypamate morpholino group. Here we are seeing the staining for tunnel, which represents apoptotic cells in green, and HB9, which is a motor neuron transcription factor, in red. Instantly, we can see the greater number of tunnel-positive cells in the trypamate group compared to the control group. On the graph on the left, we can see the number of tunnel-positive cells per embryo. We had an average of 0 in the control morpholino group versus an average of 11 in the trypamate morpholino group. What we were most interested in were the percent of HB9 positive and tunnel positive cells per embryo, meaning the percent of HB9 positive cells that were also positive for tunnel. We see that the control morpholino group had an average of 0% versus the tryptamate group, which had an average of 32%, meaning 32% of the cells that were HB9 positive for the motor neuron transcription factor were also positive for tunnel for the apoptosis. This demonstrated that tryptamate knockdown leads to an increase in apoptotic cells, specifically motor neurons, in the anterior region of the spinal cord. Here we are again seeing transverse sections of spinal cord for the embryos grown at cold temperature, but we are looking at the posterior region of the spinal cord, closer to the tail. Again, on the left, we have our control morpholino group, and on the right, we have our tryptamate morpholino group. We're seeing the same staining of tunnel, represented in green, and HB9 represented in red. Again, we can instantly see the greater number of tunnel-positive cells in the tryptamate-injected group versus the control group. Again, the graph on the left shows the number of tunnel-positive cells per section. We see this go from an average of 1 in the control group to an average of 7 in the tryptamate group. What we were most interested in, again, was the percent of HB9 positive and tunnel positive cells per section. We see this go from 0% in the control group to 34% in the tryptamate group. This meant that knockdown of tryptamate led to an increase in apoptotic cells, specifically motor neurons, in the posterior region of the spinal cord. This showed to us that there were more motor neurons that were becoming apoptotic in the embryos grown at cold temperature. This data helps us conclude that my hypothesis was supported. Tryptamate function is necessary for the prevention of apoptosis of spinal cord neurons, specifically motor neurons in animals raised at cold temperature. The rate of apoptosis also progresses from anterior to posterior regions of the spinal cord, and tryptamate knockdown accelerates the rate of motor neuron programmed cell death. My future work is to increase the number of experiments, to assess reproducibility of the data, and to examine apoptosis in the spinal cord of animals reared at warm temperatures, since the data I showed you was cold temperature. I predict that the data will be very similar to those of the animals raised in the control morpholino group, since ideally this tryptamate channel will not be active in the warm temperature. I also hope to test the hypothesis and find the mechanisms by which tryptamate-mediated calcium activity is important for the prevention of apoptosis. In this experiment, I was able to show that tryptamate presence was important in the prevention of apoptosis. What I really want to focus on is how tryptamate activity, specifically its calcium activity, is what is truly linking these motor neurons to apoptosis. These are my acknowledgments and references. Thank you so much for listening to my presentation, and a big thank you to my PI, Dr. Laura Borodinsky, and all my lab mates. Thank you to the MARC program at UC Davis and the Provost Undergraduate Fellowship for their support.